Alright, जी अस्सलाम वालेकुम everybody. How are you all? I hope that you all are doing well. Welcome to the part 2 of this video where we will be solving February March 2022 paper 1 2. In part 1 we solved question 1 till question 20, and in the part 2 of this video we are going to be looking at question 21 to question 40 inshallah, right? So now let's move on to question 21 over here. Question 21 over here says that some parts of a typical human chromosomes are most numerous than others. Which part are listed in the order from the most numerous to the least numerous? Alright, so we will discuss the options one by one. If I talk about the centromeres, right? Centromeres are connecting the two sister chromatids, right? They are one per the chromosome, right? They are connecting two sister chromatids and they are one per the chromosome. And then if I talk about the nucleotides are basically the units from which the DNA is basically made, right? DNA is made from the nitrogenous bases. So nucleotide is basically those building blocks of the DNA, right? Nucleotides are the building blocks of the DNA. And if I talk about the histones, so these over here are the histone proteins. If you talk about these spherical structures over here, these are the histone proteins, and the whole structure is known as the nucleosome, right? In nucleosome there are the turns of the DNA also included, and as well as the histone proteins are also included. And if I talk about the number of histone proteins in one nucleosome, so there are, I believe, almost 8. Yes, there are 8 histone proteins in one nucleosome, right? So basically over here this statement is incorrect. Why option A is incorrect? Why? Because the centromere is only one per the chromosome, right? And if I talk about the nucleotide, so the nucleotides are more in number than the centromeres, right? Why? Because the DNA is made up of the nucleotide. So this statement is, yeah, this option is incorrect. Then if I talk about DNA molecules, telomeres and centromeres, so we know that the centromere is one per the molecule. Telomeres are more numerous than the DNA molecules, right? Why? Because if I talk about just one chromosome over here, if I talk about just one sister chromatid over here, or the one chromosome over here, so we have two per one chromosome, right? And this is the duplicated chromosome. So over here it has gone the replication stage, this chromosome. So we have four chromosomes over here, four telomeres over here, right? And if I talk about the DNA molecule, so each sister chromatid is made up of the one DNA molecule. If we say that it is made up of one DNA molecule, right? So this statement is also incorrect. Option B is also incorrect. If I talk about the DNA molecule, if I explain you even more better, then it would be that each chromosome consists of one DNA molecule, right? Each sister chromatid or the chromosomes consist of one DNA molecule, right? Like over here, if I say that this strand is made up of this sister chromatid is made up of one DNA molecule, and this is made up of the other DNA molecule, right? Yes, option C is our correct answer over here. Let's discuss that how option C is our correct answer over here. If I talk about the histone proteins, so in the chromatin threads, you guys can see that there are a lot of nucleosomes over here, right? And in each nucleosome, there are 8 histone proteins, right? There are 8 histone proteins over here. So there may be a lot of histone proteins. So histone proteins may be most numerous, right? And if I talk about the telomere, so it is present one per the they are present two per the chromosomes, each at the poles, right? And as this chromosome has been as this chromosome is showing after the DNA replication in the S phase, then it consists of two sister chromatids as well, each with the two telomeres. So over here there are the 4 telomeres, right? And then if I talk about the DNA molecule, so DNA molecules in one sister chromatid there is one DNA molecule, in the other there is also one DNA molecule basically. सही है. Alright, I hope that you are getting it. So hence option C is our correct answer over here. Let's now discuss question 22 over here. It says that which row correctly describes a stage of mitosis. Mitosis is divided into prophase, metaphase, anaphase and telophase, right? We can remember this by PMAT, right? Prophase, metaphase, anaphase and telophase. Now they are saying that which row correctly describes the stage of the mitosis. Now let's discuss option A. It says that prophase nuclear envelope disappears in the prophase. Yes, this is correct. Centromeres replicate. No, this is incorrect as the centromeres replicate in the S phase, right? Spindle microtubules begin to form. Yes, this is also correct. But however, as the centromeres are replicated during the S phase and not during the prophase, so hence option A is incorrect. Then if I talk about the metaphase, so nuclear envelope is not present. Yes, correct. Now if we talk about the centromere, so they say move to the poles of the cell. No, this is not correct. Why? Because centromeres, if you guys remember, they connect the two sister chromatids together, right? And in the metaphase, what happens is that the sister chromatids or the chromosomes align at the equator of the cell, so the centromeres must be connecting those two sister chromatids together, right? And they must be in the center, so this is incorrect, right? And if I talk about the anaphase over here, so anaphase, in the anaphase they are saying that nuclear envelope begins to reform. This is incorrect as the nuclear envelope begins to reform in the telophase, right? Yes, this is correct that the nuclear envelope begins to reform. Now centromeres are at the maximum distance from the cell equator. Yes, why? Because the sister chromatids are pulled apart in the anaphase, right? So this is also correct. Spindle microtubules break down. Yes, this is correct because already in the anaphase the spindle fibers have shortened and the two sister chromatids are pulled apart, right? Both are at the other poles, right? So hence the correct answer for this question is option D over here. Now let's move on to question 23 over here. Question 23 says that the diagram represents a nucleotide containing the adenine. Which statement about this nucleotide are correct? So they have given us the four statements and they are saying that which statements are basically correct. The carbohydrate is pentose. Yes, this is a correct statement. Why? Because you can over here see that it is a 1 2 3 4 5 carbon sugar, right? And 5 carbon sugar is the pentose sugar. Then it says that the base contains the nitrogen. Yes, we know that all the bases A, T, G, C, which is or U, which is adenine, thymine, uracil, guanine or the cytosine, they all are the nitrogenous bases, right? They all are the nitrogenous bases. Adenine and guanine are the purine bases and thymine, uracil, cytosine are the pyrimidine bases, right? It consists of two ring structure, the purine rings, the purine nucleotides, and if I talk about the pyrimidine nucleotides, so it consists of a one ring structure, right? Base pairing will occur with the uracil. Okay, if I talk about the adenine, so adenine base pair will occur with the uracil. Yes, it is correct, right? It can pair with the thymine or the uracil as well. So yes, it is correct. Adenine is a pyrimidine. No, adenine is not a pyrimidine. It is a purine, right? It is a purine. So hence the correct answer for this question 23 would be option B. 1, 2 and 3 are the correct statements. Now let's look at question 24 over here. It says that which row is correct for the replication of the DNA molecule, right? Now for the replication of the DNA molecule, the new or the replicated strand which is made from the template strand of the DNA would be from the direction 5 prime to 3 prime, right? It would be from 5 prime to 3 prime, and then they are saying that which is that enzyme that adds the complementary nucleotides to the lagging strand. Now there are two strands over there, right? Now one is the leading strand and one is the lagging strand, right? One leading strand is formed and one lagging strand is formed, and they are saying that what is that enzyme which adds the complementary nucleotides to the lagging strand. Now so basically that enzyme which adds the complementary nucleotides to the lagging strand is basically the DNA polymerase, right? So the correct answer to this question must be option C, Now, what is the role of DNA ligase? Why DNA ligase cannot be our correct answer? Because a leading strand and the lagging strand, these both strands are known as the Okazaki fragments, and these both strands are basically joined together by the these two strands are basically joined together by the DNA ligase, right? It joins the Okazaki fragments, right? I hope that this is clear to all of you. Now question 25 over here says that some students were asked to look at the photomicrograph of a cross section of a unfamiliar material and describe what they could see. Right now in this photomicrograph, what you are seeing is that you are seeing some circular structures over here, right, which they are labeling as F. Then they are saying that the students describe the cross section of the F as follow: 1 circular, 2 a hollow tube, 3 spherical. Which descriptions of the cross section of F correctly state what the students could actually see? Now, the circular structure, yes, we know that the circular structure is seen over here, it is correct. Spherical, we cannot correctly tell that whether it is spherical or not, because we have not been given a 3D image, right? It is a 2D image, so we cannot tell over here. A hollow tube, we can also not tell that it is a hollow tube or not, right? If it would be a hollow tube, then this area would be less dark, right? So we cannot clearly tell this as well. So hence the correct option for this MCQ must be option C, 1 only. It is the best description of this cross section area, right? That F is circular. Alright, so now let's move on to question 26 over here. Question 26 says that the diagram shows the relationship between the phloem sieve tube element, xylem vessels element, and the companion cell. Right, so this is basically the chart which they are showing, right? And they are relating 1, 2, 3, 4, and 5 in it. Now they are saying that which row correctly identifies what could be represented by the number 1, 2, 3, 4, and 5, right? Number 1, number 2, number 3, number 4, and number 5. If I talk about option A over here, companion cell, right? They are saying that this is the companion cell. And they are saying 2 is the endoplasmic reticulum, and this 3 is the phloem sieve tube, right? Phloem sieve tube element, right? It is the phloem sieve tube element. Then it is saying that no nucleus is on 4, and 5 is the xylem vessel, right? 5 is the xylem vessel element. Now if I talk about the companion cell, which is a neighboring cell of the phloem sieve tube element, which basically supports the phloem sieve tube element. So companion cell, the key point which you guys have to remember for the companion cell, it has no chloroplast. The companion cell have no chloroplast, and it has more mitochondria, right? It has all other cell organelles, right? But it has mitochondria in more quantity than the other cells, right? And if I talk about the phloem sieve tube element, so phloem sieve tube element is usually a hollow, it is a hollow type of a cell, right? It is a hollow type of a cell which is having the peripheral cytoplasm, you can say, right? It is having the cytoplasm at the edges, and it has endoplasmic reticulum, mitochondria, right? But it does not have the key points which you have to remember for the phloem sieve tube element, that it does not have the ribosome, right? It does not have the ribosome, it does not have the nucleus, it does not have the Golgi body, it does not have all of these. ठीक है, Golgi body, nucleus, ribosomes. So yes, phloem sieve tube element and the companion cell, these both of the cells are having the endoplasmic reticulum. So till over here option A is correct. Now let's see that no nucleus they are writing over here, right? In the fourth they are saying that no nucleus. Now we know that the phloem sieve tube does not have any nucleus, right? I forgot to mention over here that it also does not have any nucleus. Xylem vessel elements, they are non-living, right? They are also non-living, so obviously they are also not going to have the nucleus. So hence the phloem sieve tube element and the xylem vessel element both does not have the nucleus. So hence the correct answer to this question is option A over here. I hope that this MCQ is clear to all of you. Now let's move on to question 27 over here, right? Question 27 over here says that why does an air bubble in the xylem vessel element stops the flow of the water, right? Now they are saying that the loss of adhesion, 2 is the loss of cohesion, 3 is the collapse of xylem vessel elements. Adhesion is between the water molecule and the wall, you can say water molecule and wall of the xylem vessel, the lignified wall of the xylem vessel, right? Loss of the cohesion is basically between the water molecules, right? Water to water, you can say, water to water, you can say, right? Now we must understand over here that what is basically stopping the flow of the water if the air bubble is in the xylem vessel element. So which factors are basically causing this flow of the water? Right now basically the presence of the air bubble, in the xylem vessel stops the water flow mainly because of the loss of cohesion, right? Mainly because of the loss of cohesion between the water molecules. Why? Because we know that the water moves up the xylem via the cohesion tension theory, right? We know that where the water molecules stick together, which is known as the cohesion, and are pulled upwards due to the transpiration, right? And if an air bubble forms, it will break that continuous water column, right? It will break that continuous water column. For example, this is a continuous water column was there, and suddenly this is a continuous water column, right? And suddenly what happens that an air bubble comes over here. So what will happen that it will break that continuous water column, preventing the further upward movement of the water. So hence the correct answer would be option D, which is 2 only, right? The correct answer would be option D, which is 2 only. Why others cannot be the correct answer? Like for example, why statement 1 cannot be correct? A loss of adhesion also takes place, but this is not the primary reason, right? This is not the primary reason for the water to stop flowing, right? Loss of cohesion is the main reason. And we can also say that collapse of the xylem vessel element. Yes, if I talk about the collapse of the xylem vessel element, so this can also occur, but it is not directly responsible for stopping the water flow, right? This is not the main reason here. This is not directly responsible for stopping the water flow. So hence the correct answer over here would be option D, 2 only. Let's now move on to question 28. It says that with changes to the water potential and the volume of a solution in the phloem sieve tube occurs when amino acids are moved into the sink from the phloem sieve tube. Right? This is basically over here I have drawn the phloem sieve tube elements. Just suppose that these are the phloem sieve tube elements, right? Just suppose that these are the phloem sieve tube elements, right? So they are over here saying that which changes are going to occur in the water potential as the amino acids are moved into the sink from the phloem sieve tube, right? This is the sink, and as the amino acids are being moved from the phloem sieve tube into the sink, so what is going to happen to the water potential in the phloem sieve tube? So as the solutes are moving out of the phloem sieve tube element, so the water potential will obviously become higher, right? Now let's understand the volume of solution in the phloem sieve tube. If I talk about the volume of solution in the phloem sieve tube, so you know that what happens that at the source what happens that when the amino acids are actively loaded into the phloem sieve tube, so what will happen that this will lower the water potential in the phloem sieve tube elements at the source side, right? So what will happen that water will enter over here into the phloem sieve tube elements from the surrounding xylem vessels by the osmosis, right? And this will increase the hydrostatic pressure, right? And then what will happen over here that at the sink, the phloem sieve tube element which are at the sink, what will happen that when the amino acid will be loaded into the sink, so what will over here happen that this will reduce the solute concentration in the phloem, and as the solute concentration in the phloem decreases, so what will happen that the water potential will increase, right? So what will happen that the xylem vessels which are surrounding that phloem sieve tube element at the sink, the water potential in the xylem vessels will be decreased, right? So following this, the water potential will what will happen that the water moves from the higher water potential, which is from the phloem sieve tube element at the sink, into the xylem vessels, right? So this reduces the hydrostatic pressure, maintaining the pressure flow gradient for the continuous transport, right? So hence अगर मैं summary की बात करूँ, so if I talk about the summary, so water moves out by the osmosis because the solute concentration in the phloem decreases after unloading, increasing the water potential, right? So hence the correct answer for this would be option A. Now question 29 over here says that which features of some xerophytic leaves reduces the rate of transpiration by decreasing the water potential gradient between the internal leaf surface and the atmosphere. Now if I talk about that what are xerophytes, so basically xerophytes are the plants which are adapted to live in the minimal supply of the water, so they are basically known as xerophytes. Now they are saying that which features of the xerophytic leaves reduces the rate of transpiration by decreasing the water potential gradient between the internal surface and the atmosphere. Now let's read the options one by one and let's conclude our answer. Thick waxy cuticle on the upper surface of the leaf. Now if I talk about the waxy cuticle and at the upper surface of the leaf, so what happens with the waxy cuticle, it is that it reduces the water loss by waterproofing the leaf surface, right? It is a thick layer, right? And it just causes the leaf to become waterproof, but not directly affects the water potential gradient. So this is not the correct option. If I talk about option B over here, so option B is a leaf reduced to spines with a small surface area to volume ratio. It will reduce the overall rate of transpiration. What will happen that it will reduce the overall rate of transpiration by decreasing the surface area. So it is also not involving the water potential gradient. The major cause over here is the surface area, so hence this option is also incorrect. Now if I talk about option C over here, so stomata located at the sunken pits of the leaf surface. So yes, this statement is correct. How that the sunken stomata, what will happen that the stomata which are located at the sunken pits, right? So what will happen that it will trap most of the air around it, right? It will trap most of the air around it. So what will happen that it will trap most of the moisture, around it, which is having the higher water potential, right? So what will happen that it will reduce the difference in the water potential between the inside of the leaf and the surrounding air, right? So this slows down the transpiration. So hence the correct answer over here would be option C. The moisture will be collected around the sunken pits in the sunken pits on the leaf surface, right? And what will happen that because of the high water potential of the moisture and the high water potential inside the leaf surface, what will happen that the water potential gradient will be decreased and less water will be lost, right? So hence the correct answer over here would be option C. Now let's move on to question 30 over here. Question 30 over here says that the diagram shows the cross section through the mammalian heart. Now they are saying that which chambers of the heart are represented by G and H. Very simple, you guys can see the muscular wall around the two, right? The two chambers, you can see the muscular wall around the two chambers. Over here you can see that the muscular wall is a little more thick, right? And over here you can see that the muscular wall is a little more thin. So according to me, this would be the left ventricle, right? It would be the left ventricle because we know that left ventricle requires a high pressure to pump the basically the left ventricle pumps the blood at a high pressure, right? Because it has to pump the blood to the aorta, which has to supply the blood to all of the body. So hence H is the left ventricle because it has a thick muscular wall around it, and G is the right ventricle, sorry, not atrium, ventricle, right ventricle, right? So hence the correct option would be option D. Yes, G is the right ventricle and H is the left ventricle. So hence the correct answer over here would be option D. I hope that this is clear to all of you. This is basically a transverse section. It is cut like this. For example, if this is something, right, and you are cutting it like this, right? Basically, if I tell you like this, if you are cutting it, so this is the transverse section of the mammalian heart. Now let's have a look at question 31 over here. Question 31 over here is saying that blood enters blood entering the heart from the vena cava passes through or past several structures before entering the lungs. Five of these structures are included in the list: atrioventricular node, aorta, semilunar valve, left atrium, pulmonary vein, Purkinje tissue, sinoatrial node, pulmonary artery. After arranging these five structures in the correct order of the blood flow from the vena cava to the lungs, which structure will be third? Alright, so basically they are saying that as the blood enters the heart from the vena cava, it passes through several structures before entering the lungs, right? So as it enters from the vena cava, so it must enter the right atrium, right? So you can see over here that right atrium, the option for the right atrium is not over here, right? So it must be talking about some else order, not the order of the atrium and the ventricles, right? So you can see, yes, I think they are talking about the sinoatrial node order. Like for example, first comes the sinoatrial node, right? And then there comes the AV node, then there comes the AV node, right? And then there comes the bundle of His. But the bundle of His is not given us in these options, right? So then Purkinje fibers are given, right? Bundle of His is basically between the interventricular septum, I believe, right? Yes, in the interventricular septum, and then comes the right and left branch of the Purkinje fibers. After the Purkinje fibers, it would be the semilunar valve, I believe, because as the contraction, as the electrical impulse, semilunar valve, as the contraction from the Purkinje fibers is spread along the ventricles and the ventricles will contract, it will pass the blood to the semilunar valves, right? It will pass the blood to the semilunar valve, and after the semilunar valve it will basically go to the pulmonary artery. Yes, pulmonary artery. Why? Because it has entered the vena cava first, so it will go to the pulmonary artery to the lungs, right? So lungs is also not in the option, so we will just cancel the lungs. So 1, 2, 3, 4 and 5 structures we have listed. So now they are saying that which structure will be third over here. So third over here is the Purkinje tissue, right? Third over here is the Purkinje tissue. So hence the correct answer over here is option B. I hope that this MCQ is clear to all of you. Now let's have a look at what question 32 over here is saying. Question 32 over here says that the bar chart shows the quantity of endothelial tissues, elastic tissues and the muscle tissues in the wall of the three blood vessels P, Q and R. Now this is the blood vessel P, then this is the Q and this is the R, and they are saying that this white box represent the endothelial cell, then the grey box represent the elastic tissue and a more grayer box represent the muscle tissue. Now they are saying that which row correctly identifies the three blood vessels. So the three blood vessels which we are having is the elastic artery, right? We are having the elastic artery, we are having the muscular artery, right? And we are having the vein. Now it is very clear that if we are talking about the elastic artery, right? If you are talking about the elastic artery, so in the elastic artery, the elastic tissue content must be greater, right? So I believe that P has the greatest of the elastic tissue quantity. So I believe P is the elastic artery, right? And Q, the muscular artery, let's see that whether Q is a muscular artery or a vein. So basically, the Q has a higher quantity of the muscle tissue in it, right? So I believe that Q is a muscular artery and R is obviously the vein, which has more of a muscle tissue, less of a elastic tissue and very least of the endothelial tissue. So hence the correct answer for this question 32 would be option A over here. I hope that this MCQ is clear to all of you. I think this was more of an IQ question. Now let's look at question 33 over here. It says that the maximum pressure in each of the four chambers of a healthy human heart was recorded during one cardiac cycle. The maximum pressure recorded were 3 mm Hg, 10 mm Hg, 25 mm Hg and 120 mm Hg, which values was recorded for the right ventricle. Now let's arrange these pressures from the highest to the lowest. We can say that 120 is greater than 25, then 25 is greater than 10 millimeter of Hg, and 10 millimeter of Hg is greater than 3 millimeter of Hg. If we talk about the pressures over here, so the pressure of the left ventricle is the highest pressure, right? Then comes the pressure of the right ventricle. Then comes the pressure of the left atrium, which is usually higher than the pressure of the right atrium. So basically they are talking about the pressure of the right ventricle. So the pressure of the right ventricle would be 25 millimeter Hg. So hence the correct answer for this MCQ would be option C over here. I hope that this MCQ is clear to all of you. Now let's move on to question 34. Question 34 over here is showing the cardiac cycle graph, right? The graph shows the pressure changes in the three structures of the right side of the heart during the cardiac cycle. Alright, so it is basically a pressure time graph what is represented by the line label T. Alright, so let's discuss that what each line represent over here. This line over here which they are indicating as the T, as it is the right side of the heart, so it must be the pulmonary artery. Pulmonary artery. If they were talking about the left side of the heart, then also this is the artery, but if they were talking about the left side of the heart, so it would be aorta, but for now they are talking about the right side of the heart, so it would be the pulmonary artery, right? And then this line over here, the dotted line, this one, it is representing the atria, the right atria, right? And over here this whole line, which I can highlight over here as this whole. So this is representing the right ventricle. Alright, so what would be the correct answer over here? The correct answer over here would be the pressure changes in the pulmonary artery. Option A would be our correct answer over here. I hope that this is clear to all of you. Alright, so if we revise more of this graph, so it would be like, for example, this part over here would be called the atrial systole, right? This part over here would be called as the atrial systole. Then this part over here would be called as the ventricular systole, right? Ventricular systole. And then this whole part would be the diastole, right? So yeah, that was about question 34 over here. Now let's move on to question 35, right? Question 35 over here says that which statement helps to explain why there is no cartilage in the walls of the bronchioles. Now we know that there comes the trachea, then there comes the bronchus, and then the bronchus divides into more branches which are known as the bronchioles, right? Now they are saying that why the no cartilage is present in the bronchioles. Cartilage would make the bronchioles too narrow. No, incorrect. Gases must diffuse across the walls of the bronchioles. No, gases only diffuse across the alveolus. Smooth muscles is sufficient to support the wall of the bronchioles. The bronchioles wall do not need to change the shape. No, this is not correct. Cartilage are not needed to change the shape basically. I think the correct answer for this question 35 would be option C, which is that the smooth muscles is sufficient to support the walls of the bronchioles, right? Why the C-shaped cartilages are present in the trachea? To prevent the collapse of the trachea under high pressure, right? So basically cartilages are there to support the walls of the trachea, bronchus, right? And but for the bronchioles, no cartilage is present. Why? Because the smooth muscle is sufficient to support the wall of the bronchioles. That's why, right? So I hope that this is clear to all of you. Alright, so now let's have a look at question number 36 over here. It says that the photomicrograph shows a cross section of part of a gas exchange system of a mammal. What is shown by the line labeled V? So over here you must know the knowledge of the photomicrographs. You must know how to identify the photomicrograph. If I tell you, so there is a trachea, then there comes the bronchus, right, and then there comes the bronchioles, right, and then you have the alveolus. So if you guys see over here, so this structure over here are the alveolus, right, and this structure, which is this structure, you guys can see it has a ciliated epithelium which is wavy in nature, right. So the ciliated epithelium which is wavy in nature is basically a characteristic of the bronchioles, right. So basically this V is representing the diameter of the bronchioles. So hence the correct answer for question 36 over here is option B. I hope that this MCQ is clear to all of you. The gas exchange micrographs and the photomicrographs are very important. You should know how to identify each and every structure, alveolus, bronchioles, bronchus, trachea in the photomicrographs, right. Alright, so question 37 over here says that which feature of the disease cholera decreases the spread of the pathogen Vibrio cholera. So basically cholera over here is an infectious disease which is caused by the bacteria Vibrio cholera, right, which it primarily spreads through the contaminated water and the food, right. So the rapid, basically they are saying that what causes the decrease in the spread of the pathogen, right. So we want to decrease the spread of the pathogen Vibrio cholera over here, right. So if I talk about the options one by one over here, so let's look at this. Immunity to cholera after vaccination is short lived, lasting less than 2 years after vaccination in 50% of the people. A short lived vaccine basically means that people remain susceptible, which usually increases the spread of the cholera rather than decreasing it, right. So this statement is also incorrect over here. Then it says that up to 98% of the people infected with the Vibrio cholera are symptomless carriers, right. If they are asymptomatic, right, if they are symptomless, so they can unknowingly spread the bacteria, right, which increases the transmission rather than reducing it. So yes, this statement is also incorrect. Then now let's see statement C over here. Statement C over here says that cholera rapidly kills up to 50% of the people with these symptoms if they are not treated. Alright, so if I talk about is that if an infected person, for example, if a person was infected and if he dies quickly, then they have less time to excrete the large amount of Vibrio cholera in their feces, right, which is the main route for the transmission. So this reduces the pathogen chances of contaminating the water supplies and spreading to the new host, right. So the correct answer over here would be option C, that cholera rapidly kills up to 50% of the people with symptoms if they are not treated. So it will decrease the spread of the pathogen Vibrio cholera, right. So I hope that this MCQ is clear to all of you. Alright, so now let's move on to question 38. It says that which row shows how penicillin kills the bacteria. Penicillin is basically an antibiotic, right, and it helps in killing the bacterial cells. So basically they are saying that which row shows how penicillin kills the bacteria, process which is inhibited by penicillin and its effect on the bacteria. So during the initial stage where the bacterial cell wall is being made, right, this is the bacterial cell wall which is during the initial stage before it is being made, right. So this is the peptidoglycan molecules, right. We know that the cell wall of the bacteria is made up of the peptidoglycan, right. So basically the cross links are formed between these peptidoglycan, so that these peptidoglycan molecules can link together to form a continuous cell wall of the bacterial cell, right. So now what happens over here is that due to the penicillin basically inhibits, the role of the penicillin is that it basically inhibits the formation of the peptidoglycan crosslinks over here, right. It inhibits these, right. It does not allow them to form over here. So what is the effect of this on the bacteria? That water will enter the bacterial cell and the cell will burst, right. If water will enter into the cell, because no crosslinks are there, right, so water will enter into the cell via osmosis and the cell of the bacterial cell will be burst, right. So basically penicillin kills the bacteria by interrupting the by interrupting the formation of the cell wall. By interrupting the formation of cell wall. So hence the correct answer for this MCQ would be option A over here. I hope that this MCQ is clear to all of you. Now over here in question 39 they are saying that in the hybridoma method what is grown by the cell culture to produce the. Now in question number 39 over here they are saying that in the hybridoma method what is grown by the cell culture to produce the monoclonal antibodies. Now we know that what are basically the hybridoma cells. Hybridoma cells are basically made by combining the plasma cells and the myeloma cells, right? And they both combine together to form the hybridoma cells, right? So now basically in the what happens in the production of the monoclonal antibodies, that these hybridoma cells, the clones of the hybridoma cells are basically grown by the cell culture to produce the identical monoclonal antibodies, right? So the correct answer over here would be option B, that clones in the hybridoma method, the clones of the hybridoma cells are basically grown in the cell culture to produce the monoclonal antibodies, right? I hope that this MCQ is clear to all of you, that we are growing the clones of the I hope that this MCQ is clear to all of you, that we are basically growing the clones of the hybridoma cells in the cell culture to produce the identical monoclonal antibodies, right? Now in question number 40 over here they are saying that which type of immunity does a baby have at birth? Artificial, active natural, passive natural, or the passive artificial, or the passive natural. Alright, so before we discuss the answer, let's now first have an idea that what basically the active and passive immunities basically are. So if I talk about the active immunity over here, so active immunity is the immunity which is basically gained due to the antigen, right? Which is gained due to the antigen. Now the antigen can enter into the body naturally from the environment, or it can be injected artificially through the vaccines as well, right? And then the other type of the immunity we are having is known as the passive immunity. Right now the passive immunity is basically gained by the antibodies. Now that antibodies can come in your body naturally as well as artificially. Artificially they can enter your body through the anti-serums, right? And naturally they are found in the breast milk, right? And the mother to the fetus, the antibodies basically pass from the mother to the fetus via the placenta, right? So they are saying that which type of immunity does a baby have at the birth, right? Baby have at the birth, so baby have at the birth the natural passive immunity, right? The natural passive immunity is the immunity which the baby has at the birth. How does it have the natural passive immunity? Because the antibodies are passed from the mother to fetus via the placenta. So hence the correct answer over here is option D. Alright, so that was the end of this paper of February March 2022, paper 1-2. I hope that you guys enjoyed solving this paper with me and all of the MCQs are clear to all of you. If you guys do find these solutions of the MCQs helpful, then do consider subscribing this channel and remember me in your prayers. I will see you guys in the next past paper video. Till then, have a good day and keep working hard for your upcoming examinations. The very best of luck to all of the students from my side.